Thank you everybody for coming. Thank you also Rodrigo, Kelly again, and Virginia for organizing this meeting and inviting me to be here. And mainly thank you to Professor Dimas to give me the opportunity to be part of the team of cell therapy of this institution, mainly involved with CAR T cells and other aspects related to that. So I'm going to talk about T cell, of course, expansion protocols. I'm not going to, my goal here is to give you some principles in terms of cell biology, how we use the pathways of activation, proliferation, and to design our experiments and to try to produce or polarize T cells as we want to have to treat our patients. So if I give you the message of the principles of cell biology, T cells, I'll be happy at the end. Then I'm going to show you some clinical trial results that comes from my boss at NIH, and that will be everything that I'm going to explain for you. This is my conflict interest to disclose. I have nothing. I don't receive money for anybody. And so I'd like to start with this classical platform that belongs to a stem cell transplantation scenario. It's a very rich environment of immunology principles and aspects. So basically you have two players here. At one side you have the health donor, and the other side you have the patients that needs to be treated normally with health treating disease. And for a long, long time, 30 years, this has been the only opportunity to treat the majority of patients that we receive in our hospital. And now we are moving toward cell therapy, mainly genetic modified cells. But until there, just this used to be our option, and basically this is a very summarized chart. So if my donor cells engraft, I can have successful of this treatment, what can be introduced in survival, and graft versus leukemia effect or tumor effect. But this effect and or reaction can be very strong and harmful for my patient, and this can be translated in graft-versus-host disease, what can promote mortality. But if my transplantation doesn't work, I can have graft failure and relapse. So this is very basically, but if I look on my T cells and APCs, I can see that I receive CD4 and CD8 from my donor. Where's my cursor? It's here. And with APC from the donor. That also you'll be interacting with host APC, and this APC will present the antigens for my donor cells, CD4 and CD8, and these cells will be reactive against the host, or in this case, the patient. And you can have skin, here, gut, and liver reaction, what we call acute graft-versus-host disease. This can kill my patients, okay? So this is the basic scenario, just to understand. And when I dissecate, or when I try to see the details of the players of this scenario, I can find that acute graft-versus-host disease are mainly related to type 1 immune response. So this is my TH1, T helper 1, or TC1 cells, CD4 and CD8. So it's very inflammatory environment. I have a lot of interferon gamma. I have a lot of TNF alpha in this scenario. But on the other hand, if I have a TH2 response, I have more IL-4, IL-5, and I have a reduced inflammatory cytokines process, and this could give some GVL for my patients and not a very strong GVHD, so this could be translated in survival. If I can separate both processes, I will have the perfect scenario to give to my patients. So in allogeneic transplantation settings, if I promote a TH2 response, that could be traduced in improved survival, okay? So that's the principle and the rationale that I bring from bone marrow transplantation. But how can you do that? So now we start the cell biology process. We talk about TCR, co-receptor, and everything. Let's try to understand that a little bit in details. Here's the famous guy, TCR. So this is our CD3 molecule and receptor. So he's related with nuclear factor of activate T cells. So if I stimulate just my CD3, my T cells will be partially stimulated and activated. So for that, I need to activate my some co-receptors here represented by CD28, what I call signal 2. TCR is my signal 1. So if I activate signal 2, I will have the NF-kappa beta pathway activated and also my activated protein 1. So I will have the full process of activation done, and this cell is ready to start to proliferate. Okay? So that's why you use CD3 bar CD28 beads in T-cell cultures. That's the principle. But once that is done, I have a lot of new receptors for chemokines and cytokines that are overexpressed in my cell surface. And in this process, I can activate it, or in fact, the right to expression is I can use these receptors to polarize my cells. So if I use IL-12, I can have a more TH1, TC1 cells. If I use IL-4, I can have my TH2, TC2 cells, and then go forward with that principles. So this is my signal 3. So I can give you a recipe based on that principle. So is that what I have in my mind every time when I design experiments? And basically, based on the type of cells I want. So let's see the first on the top. I have some naive cells. You can see here on your right, these cells are CD62L and CD45RA, so that means the cells are naive cells. It's very undifferentiated cells. And if I put the cells in culture with interferon alpha and IL-12 together with CD3, CD28, and I expand that for 6 days, 10 days, 12 days, I suppose that I have at the end of my culture a TH1 cells expanded. So if I look at the transcription factor, this will be mainly TBAT, what is related with this type of response. But on the other hand, or conversely, if I use IL-4, as you can see in the bottom here, I have more TH2 response. So this is a GATA3 mediated cell differentiation. And in the middle, I put here both TH17 and TH2-reg, because they share the same cell at the beginning, and all are dependent of TGF-beta process of expansion. So there are some plasticity here between both cells. If I express ROR gamma T, RO gamma T, I will have TH17, and FOXP3 is related to REG. So the main player here, what's very interesting to see is the alpha HIF1. So when I have a hypoxia process in the inflammatory environment, I have an overexpression of alpha HIF1, and this can block my FOXP3, and I can promote a TH17 response. This can happen in the autoimmune disease, but this also can happen in a microenvironment tumor where you have hypoxia process, and this could be a answer of our immunological response to overcome this hypoxia and this tumor microenvironment process. We know that doesn't work very well every time, because these cells, when it's chronically stimulated, you can have some PD-1, CTLA-4, KLGR1, and many other markers of exhaustion or dysfunctional process, and these cells, in fact, can't work. That's why the PD-1 blockade works very well. So just moving a little bit forward, but I want to stay a little bit close, because I also want to say if you stimulate these cells very harmful with a lot of high dose of IL-2, you can promote T effector memory cells. But if you don't do that very strongly, you can have central memory cell. I'm not going to talk about stem cell memory. It's another chapter, is much more detailed. But just to know that all these variables are together, interleukins, intensity, dose, and everything, to see or to have the cells that you want to work with. Okay? So all this immunologic process has a metabolic background. So that's very interesting to look. Sometimes a little bit boring because a lot of pathways to study, to read about. But I'm going to talk about mTOR. This is very non-transcription. MYC and HIF1 alpha, I already talked about that. So if you have all this process of activation, you will be upregulation functionally of mTOR, and you can see the cells that the synthesis of nucleotide, proteins, fat acids, and all the metabolic process of the cells are preparing the cells to divide and proliferate. So basically is that what happened in the metabolic process. We can measure that with many different methodologies to show that our cells is polarized to a metabolic background of proliferation division. There's many other, like seahorse, it's a very interesting machine that you can use, and many other ones. So normally you can use that. So if I look to mTOR, I have a specific drug for that, rapamycin, and it's not a full blocking cell drug. It's partially inhibitor of mTOR. I can maybe explore this pathway to expand my cells. It's okay. On the other hand, I just put here the calcineurin inhibitor, cyclosporine, because it's very common for us that work with bone marrow transplantation. We use a lot that, but in fact, calcineurin inhibits the NFAT, and the rapamycin you inhibit mTOR. Okay, just to differentiate both cells, both drugs. If I block mTOR, the consequence of that will be the cell cycle arrest, normally G1 phase, some in S phase. I have a low synthesis of kinase inside the cells, and also I have less protein translation. So this will be the global effect in my cells. But if I put RAPA cells, if I put rapamycin in my culture, this could be anti-proliferative and I could kill all the cells. But no, that's not true. If I use that in my culture for TH1 and TH2 environment, I can see here I have a dose curve dependent response and many different concentrations for both condition, and I have cells proliferation. So it's possible to use that in the clinical grade scenario. And but I have to see how can this can be done, because look, if I don't stimulate my cells with cytokines, I don't have any proliferation. Look here, cells culture with RAPA is inferior to cells don't culture with RAPA. But if I put cytokines, here I put, IL-12 and interferon alpha, I can rescue some cells, and these are the cells that I'm gonna work. So the process here, when I culture cells like that, and use that in vitro too, but in vivo experiments, you can see some advantage in cells that are not cultured with RAPA. For instance, here I can see the, this is the engraftment of the cells culture with rapamycin here, and you can see how this proliferation is superior to cells doesn't culture with rapamycin. Okay, so seems to be that CD4 and CD8, these cells that overcome T-cell rapamycin resistance has improved proliferation when compared with cells don't culture with rapamycin. Also, if I look this process during many different cell cycles, I can see that cells with RAPA are more frequent. You have more cells than the cells don't culture with not culture with rapamycin. Okay, this is a CFC label experiment. It's a very common experiment for proliferation. But also, what is interesting, this is a young, it's a 2006 data, so that is very well known. Here, day 2, normally I see that in my cultures with day 6 and day 8, but you have more undifferentiated cells with rapamycin. So we have CD62L positive cells when compared with cells that are not cultured with RAPA. So it seems to be that you can have a more proliferative cells with a more memory profile. So what would be the rationale in general? So the general idea here, I induce the starvation of my T cell with mTOR blocking. I stimulated these cells with cytokines, and the cells need to proliferate. And what the cells do, there are 5 explanations for that, but the main one is autophagy. So as the mTOR is not a 100% blocking process, you can escape some signal from that, and you can have autophagy process, so the cells can use the own structures of the cells to produce energy, and at the same time to survive. But that can change a little bit all the pathways in terms of apoptosis and proliferation, and seems to be that these cells could have an advancement to another cells. Okay, so in that I can use my environment, cytokines, to produce Th1, Th2, or even Tregs. Here, what Dan Fowler has been doing is to work in a transplantation scenario, but if you think about genetic modified cells, I could polarize my cells and then put my CAR and see if that could influence the response of a CAR or of a TCR engineer, for instance. So this could be also translated for this process. I'm not doing that yet, I plan, but at this time I'm not going to show anything in this way. This is I think the most important trial from Dan. So he decided to use CD4 T cells, stimulated CD3, CD28, and the IL-4 and rapamycin, cultured their cells and treated the patients. At the end, you have the majority of cells are GATA3 because IL-4, but you have also some cells with T-beta, so it's a 2-for-1 ratio process. And this is the protocol design that he used to treat these patients. Flu-cy as conditioning regime, sirolimes here, cyclosporin. This is the PBSC infusion, so is the standard stem cell to the patient. And then 14 days after day 0, they use RAPA product here. So I'm gonna move a little bit fast. I'm worried about the time. And just to show that it's safe to use this approach. So to RAPA conversion of mixed chimerism, so we have here day 14 post-transplantation. This is for T-cell chimerism. When infused into RAPA, you have the improvement of donor chimerism of my patient. So you can see all the curves going up, what is very interesting, just one patient not. And this is my myeloid cell chimerism. It's important to say and to highlight that it's a single arm study. We didn't compare that with DLI, standard DLI. So this is the GVL response before transplantation, 28 days after, D plus 28, and then D plus 60. You can see the mass here, mediastinum of the patient, and reduce the tumor mass. And the general results that you have for these patients. This is for low intermediated group. It's quite homogeneous. It's lymphoma and CAL. So Diego showed that list. So it's almost impossible to do a trial just for one subtype of lymphoma. You have to put everything together. But this is not very high risk patients. You have just 3 patients with progressive disease. These are 16 cohort patients. This one here, it's myocardial ischemia, and the other one, he's thromboembolism, lung thromboembolism, if I'm not wrong. But all the other patients are in complete remission. So this is 4 years follow-up. But here I have the high-risk patients. So this is very heterogeneous because I have AML, lymphoma, and many other disease together here, CML too. And if you look here, I have 26 patients, 15 died, 11 patients survived, okay? Just in general. So the conclusion of this study here is ex vivo manufactured allogeneic to RAPA cells can be combined with in vivo sirolimes. We put sirolimes in the conditioning regime and associate to RAPA cells to provide a platform for the safe implementation of low-intensity allo HCT. So I give a low-intensity HCT for my patient. What is I have low toxicity because I have a better cell to infuse later. Preemptive to RAPA cell infusion after low-intensity allogeneic HCT, therefore represents a suitable platform for further studies, as I stated here, not only for transplantation, but also probably it's possible to move forward in a genetic modified agenda for this platform. This is another study now work with autologous. So here is to produce TH1, TC1 cells. Look here, I have interferon alpha. You can use IL-12 too. IL-12 is much more expensive. They decide to use interferon alpha, what is also it's FDA approved. So this was the final design of the study, high risk multiple myeloma and induction therapy, auto transplantation 4 days after you can give RAPA cells for this patient. This is a phase 1 trial, escalonated cell dose will be performed. And for relapsed multiple myeloma, also then has a protocol. And here the idea is to give a 7 day of pento testing with cyclophosphamide regime, what he called PC regime, and then TH1, TC1 RAPA, 1 cycle, 6-day stop, second cycle. I check the disease response, and then you can see if you go to 14-day of PC regime, or you can stay with 7 day, depend on the response, complete remission, partial remission, or stable disease, or progressive disease. Okay, why then use pentostatin here? This is quite interesting. In fact, it came from this study here. These patients with kidney cancer. What we know that they use rapamycin to treat these patients, but the tumor cells can overcome the blocking of mTOR by rapamycin and became resistant, and then there are no response for rapamycin with patients with cancer. So he decided to use this patient to see if the RAPA could be, because we use a very high dose in vitro with RAPA cultures. It's very different what you have in blood when you treat patients. So basically he used a 20-day cyclophosphamide and pentostatin, and then he get the cyclophosphamide dose according to lymphocyte counts and adjust the dose for that. And at the end, look here, this is the neutrophils of the patients. 10 patients treated here, and 9 patients died, in fact. But this study was useful to see that I can maintain my granulocytes higher, and at the same time I can lose my lymphocytes, deplete lymphocytes. So it's a specific condition regime to T-cell depletion. So it's quite interesting too. So I'm gonna end with this slide here just to show that the basic platform, what is very intensive in terms of chemo and radiotherapy, now is moving toward a more immunological or immunotherapeutic platform. So I continue to have my health donor, the patient that I need to treat, and I can do allo transplantation with all the positive and negative aspects. But these cells now of health donor, and including also the patient in the autologous platform, I can genetically modify the cells. I can use, I didn't talk about Treg, but you can use rapamycin for that. You can polarize the cells, what I show you, or you can also to merge these platforms and see what kind of results in preclinical process you have. And this is another one. There's 2 weeks ago, a paper published in JCO. People in Europe create a bank of virus-specific T cells, and they're using that for high-risk transplantation patients. It's very nice. It's really impressed with these studies. And also, you can take advantage of what you have in terms of TCR profiling. Dr. Levine has shown here how the clones after infusion cells and proliferation happen. This is something that you can learn a lot about because you can see specific repertoire T cells that can be useful for treat patients. And also immunologic monitoring. Normally now we have flu with 13, 14 colors and many other devices that you can put in the game here to help us to understand what's happened with our patients. So thanks for, this is Dan. So this is the guy that think about the RAPA cells. Dr. Levine also is part of this process at the beginning. And the other side is Dr. Grass. Now my boss. He's the branch chief and it's very nice to have him as a boss now. Also, I'd like to say thank you for DTM, it's our transfusion medicine department. The imaging and flow cytometry core people are very helpful, and they're really very important to do all these experiment. And also a special thank you for people in Ribeirão Preto. I'm a professor, Ribeirão Preto, no? Ribeirão Preto. I'm a physician from a government unit, so once I go to US to study, I don't have another person to work in my place. So bone marrow transplantation unit, mainly Belinda, Professor Belinda, has done all the efforts to help me with this process, and I'm very grateful for that. So thank you so much. Sorry for not showing original data, but just to show the principles, what you have been doing and evolved at NIH. Thank you so much. Have a nice day.